So hello guys, welcome back in another video tutorial from microbiology with Charnik. So in this video tutorial we will going to talk about how antigen is processed that will going to be presented with MHC class 2 molecule to helper T cell. Okay, so first of all I again want to help you to remember the mnemonic that I have said in my previous classes, that is MHC is like our father. They will going to serve our family's problem first, then they will look on the external problems. Okay, so MHC class 1 molecule will going to take care of endogenous antigen, and MHC class 2 molecule will going to take care of exogenous antigen. Clear? So now exogenous antigens. So now first thing is, exogenous antigen means what? Exogenous antigen means antigens which are coming from outside, right? So what will be the first job for a cell that will going to express MHC class 2 molecule? So the first job will be to somehow take that exogenous antigen, right? So they will take that exogenous antigen most of the cases by endocytosis. We will going to talk about this. Okay, and it will be processed by exogenous pathway. Okay, and presented on the membrane with MHC class 2 molecule. Now you tell me about the name of the cell which will going to express MHC class 2 molecule. So obviously the cell name is antigen presenting cell. I have told you. If you have not watched the MHC class 2 molecule video, then please go and watch. This is available in this MHC playlist. Okay, so MHC class 2 molecule will be expressed on antigen presenting cell. Okay, and MHC class 2 molecule will going to interact with T cell receptor present on the surface of the helper T cell. Clear? So now let's move on the next page. So here one important thing that I can also mention this here, that the helper T cell will contain CD4 marker, right? And cytotoxic T cell will contain CD8 marker. Please remember it very carefully. Okay, so now I have told you, when I have talked about the endogenous pathway, that is, you can prevent the expression of class 1 presentation by any kind of protein synthesis inhibitor, right? So now remember, you cannot use any kind of protein synthesis inhibitor if you want to prevent the expression of the MHC class 2 molecule. Okay, so because class 2 presentation can occur either live or replication incompetent virus, so protein synthesis inhibitor had no effect, indicating that the new protein synthesis is not a necessary condition for class 2 presentation. But in case of class 1 presentation, presence of virus, that means the target cell must be infected with live virus, otherwise it will not going to express class 1 MHC molecule. Clear? And that is the idea, because virus infected cell or cancer infected cell are usually our endogenous antigen. Okay, when I will going to take a complete playlist on antigen, you will get a clear idea about that. Okay, so now how we can inhibit class 2 presentation if we want to, how we can do this? So remember, class 2 presentation can be inhibited by chloroquine, and it is a inhibitor that can inhibit the endocytic processing. Okay, and I have told you that first of all antigen presenting cell will going to take the extracellular antigen by endocytosis, right? So if you use chloroquine, then it will going to be very difficult for that cell to express class 2 molecule. Clear? That is the idea. So now I am okay, no problem. Now extracellular material first of all have to enter inside the APC or antigen presenting cell. And I hope you can remember the classification of antigen presenting cell that we have discussed in case of our MHC class 2 molecule video, that is professional antigen presenting cell PAPC and our non-professional antigen presenting cell, right? So professional antigen presenting cell can take up the particulate material, that is your exogenous antigen, by simple phagocytosis or cell eating. Okay, and I have already discussed about the cell eating when I have talked about the complement system, right? That phagocytosis is the process by which antigens are eaten by antigen presenting cell, mainly macrophages, right? So now let's see how it is, the entire thing is going on. So this is the diagram for MHC class 2 molecule. Okay, so first of all, the so I hope you know that antibody is Y shaped. Okay, and this is FAB portion, that is antigen binding portion. So here antigen will bind. Clear? Now first thing is, this is the step number 1, where the antigen will be coated with vesicles, and it is made up of clathrin. Okay, because remember, whenever something is going to be endocytosed by the endocytosis, a vesicle will cover that particular particle. Okay, so now after that, in the second step, the vesicle will be released. Okay, and it will be transferred to early endosome. Okay, where the cleavage of, and so here these are the antigens, so the cleavage of the antigens will occur. Okay, and they will be converted into peptides, and I have mentioned the length of the peptide in case of MSC class 2 molecule, that is 13 to 15 amino acid. Okay, now simultaneously another thing is going on in case of endoplasmic reticulum. Okay, so that is the formation of MHC class 2 molecule. Remember, MHC class 2 molecule consists of alpha 1, alpha 2, the arrangement will be just like that: alpha 1, alpha 2, beta 1, beta 2 domain, right? So along with that, invariant chain will also formed, right? Now let's try to remember, in case of MHC class 1 molecule, the MHC class 1 molecules are completely synthesized in rough endoplasmic reticulum lumen, right? And it is synthesized in endoplasmic reticulum. Okay, along with invariant chain. Okay, their invariant chain will going to play a very important role in after some time. So after that, this MHC class 2 molecule along with invariant chain will further modified and go through the Golgi complex. Clear? Now, two important point that I used to confuse during my graduation days, that is the part of the Golgi complex which is towards the endoplasmic reticulum is known as cis form or convex form or forming phase. Remember, that is cis Golgi part. Okay, and the part of the Golgi complex which is not towards endoplasmic reticulum is known as transform or concave form or maturing phase. Clear? So after that, this MHC class 2 molecule along with invariant chain will be surrounded by a vesicle, as you can see. Okay, and invariant chain will be at the place of peptide binding group. Okay, and I hope you can remember that peptides binding groups in case of MHC class 1 class 2 molecule are formed by alpha 1 and beta 1 domain. Okay, so now after that, remember, inside the vesicle the pH is acidic. Okay, not in that case only, but in every cases. Okay, so that is why this acidic condition will facilitate the digestion of this invariant chain. Okay, and as a result only a clip will remain. Okay, and this clip will remain in the peptide binding group. Okay, and this is the situation of late endosome. Okay, now what will occur, this peptide containing vesicles and this one will going to fuse. Okay, and as a result now peptides are also there, but remember this peptide binding group is already blocked with a clip, right? So the clip have to be removed so that the peptides can bind at the peptide binding group, right? So now remember here two protein will going to work. One is HLADM, another one is HLADO. Okay, so HLADM here will going to act as a positive regulator. How this HLADM will tell that clip that please buddy remove, please leave your place for the peptides. They are waiting to be presented for CD4 T cells. Okay. On the other hand, HLADO will tell that clip you will not move. Okay, you will remain there where you are now. Okay, so here how HLADO is acting as a negative regulator. Okay, so remember that if HLADM is going to work, then antigen will be on the peptide binding group, otherwise not. After that this vesicle will be released and now MHC class 2 molecule is ready for presentation to helper T cell. Okay, so I hope this is very clear for you, right? For a quick recap, first of all, endocytosis will occur, and as a result, the antigen containing antibody will be covered with a vesicle. It will be transferred to the early endosome where cleavage of antigen will occur. This is very important MCQ question for your any kind of entrance exam or any kind of job exams. Okay, where your immunology is present, and after that here simultaneously in the endoplasmic reticulum invariant chain is formed and MHC class 2 molecule is formed. Now it will be transferred through the Golgi complex and it will be then covered with a vesicle. Okay, now inside the vesicle due to the acidic condition the invariant chain will be degraded. Okay, and only a small part will remain. This small part will be known as clip. Okay, and this is the late endosomal phase. After that fusion of peptide containing vesicle and MHC class 2 molecule containing vesicle will occur, and as a result now we are in a situation where we need someone to tell that clip to be removed or to leave its place for peptides. Okay, so here HLADM will going to do that work as a positive regulator, and if in place of HLADM HLADO is present, then this HLADO will work as a negative regulator. Okay, and as a result clip will remain there. Okay, and as a result antigen cannot bind. Okay, and if HLA-DM is present antigen will bind and then it will be released. Okay, now MHC class 2 molecule along with the antigen is ready to be presented to helper T cell. Okay, or CD4 marker containing T cell. That is the entire concept of MHC class 2 molecule antigen presentation. So I hope this video will be helpful for you, and if it is, please hit the like button, share with your friends and subscribe to my channel to get more videos like that. So along with that, we have completed our playlist on major histocompatibility complex. Now I want to tell you something that after that there will be no immunology playlist immediately. Okay, because of some unavoidable reason, but still if you are having any kind of problem in immunology then you can discuss with me in the comment box, or if you are having any kind of problem in the concept then I will surely try to make some video on that, but detailed video in a chronological order will not be possible for me now. Okay, so I apologize for that. So thank you for listening to this class.